Genetic Effects of Radiation by Isaac Asimov and Theodosius Dobzhansky. Mutations, sudden change. Shifts in chromosome combinations, with or without crossovers, can produce unique organisms with characteristics not quite like any organism that appeared in the past, nor likely to appear in the reasonable future. They may even produce novelties in individual characteristics, since genes can affect one another, and a gene surrounded by unusual neighbors can produce unexpected effects. Matters can go further still, however, in the direction of novelty. It is possible for chromosomes to undergo more serious changes, either structural or chemical, so that entirely new characteristics are produced that might not otherwise exist. Such changes are called mutations. We must be careful how we use this term. A child may possess some characteristics not present in either parent through the mere shuffling of chromosomes, and not through mutation. Suppose, for instance, that a man is heterozygous to eye color, carrying one gene for brown eyes and one for blue eyes. His eyes would, of course, be brown, since the gene for brown eyes is dominant over that for blue. Half the sperm cells he produces would carry a single gene for brown eyes in its half set of chromosomes. The other half would carry a single gene for blue eyes. If his wife were similarly heterozygous, and therefore also had brown eyes, half her egg cells would carry the gene for brown eyes and half the gene for blue. It might follow in this marriage, then, that a sperm carrying the gene for blue eyes might fertilize an egg carrying the gene for blue eyes. The child would then be homozygous, with two genes for blue eyes, and he would definitely be blue-eyed. In this way, two brown-eyed parents might have a blue-eyed child, and this would not be a mutation. If the parent's ancestry were traced further back, blue-eyed individuals would undoubtedly be found on both sides of the family tree. If, however, there were no record of, say, anything but normal color vision in a child's ancestry, and he were born colorblind, that could be assumed to be the result of a mutation. Such a mutation could then be passed on by the normal modes of inheritance, and a certain proportion of the child's eventual descendants would be colorblind. A mutation may be associated with changes in chromosome structure sufficiently drastic to be visible under the microscope. Such chromosome mutations can arise in several ways. Chromosomes may undergo replication without the cell itself dividing. In that way, cells can develop with 2, 3, or 4 times the normal complement of chromosomes, and organisms made up of cells displaying such polyploidy can be markedly different from the norm. This situation is found chiefly among plants and among some groups of invertebrates. It does not usually occur in mammals, and when it does, it leads to quick death. Less extreme changes take place too, as when a particular chromosome breaks and fails to reunite, or when several break and then reunite incorrectly. Under such conditions, the mechanism by which chromosomes are distributed among the daughter cells is not likely to work correctly. Sex cells may then be produced with a piece of chromosome or a whole one missing, or with an extra piece or whole chromosome present. In 1959, such a situation was found to exist in the case of persons suffering from a long-known disease called Down syndrome. Each person so affected has 47 chromosomes in place of the normal 46. It turned out that the 21st pair of chromosomes, using a convention whereby the chromosome pairs are numbered in order of decreasing size, consists of 3 individuals rather than 2. The existence of this chromosome abnormality clearly demonstrated what had previously been strongly suspected, that Down syndrome originates as a mutation and is inborn. Most mutations, however, are not associated with any noticeable change in chromosome structure. There are instead more subtle changes in the chemical structure of the genes that make up the chromosome. Then we have gene mutations. The process by which a gene produces its own replica is complicated, and while it rarely goes wrong, it does misfire on occasion. Then, too, even when a gene molecule is replicated perfectly, it may undergo change afterward through the action upon it of some chemical or other environmental influence. In either case, a new variety of particular gene is produced, and, if present in a sex cell, it may be passed on to descendants through an indefinite number of generations. Of course, chromosome or gene mutations may take place in ordinary cells rather than in sex cells. Such changes in ordinary cells are somatic mutations. When mutated body cells divide, new cells with changed characteristics are produced. These changes may be trivial or they may be serious. It is often suggested, for instance, that cancer may result from a somatic mutation in which certain cells lose the capacity to regulate their growth properly. Since somatic mutations do not involve the sex cells, they are confined to the individual and are not passed on to the offspring. End of section 5. Spontaneous mutations. Mutations that take place in the ordinary course of nature without man's interference are spontaneous mutations. Most of these arise out of the very nature of the complicated mechanism of gene replication. Copies of genes are formed out of a large number of small units that must be lined up in just the right pattern to form one particular gene and no other. Ideally, matters are so arranged within the cell that the necessary changes giving rise to the desired pattern are just those that have a maximum probability. Other changes are less likely to happen, but are not absolutely excluded. Sometimes, through the accidental jostling of molecules, a wrong turn may be taken, and the result is a spontaneous mutation. We might consider a mutation to be either good or bad, in the sense that any change that helps a creature live more easily and comfortably is good, and that the reverse is bad. It seems reasonable that random changes in the gene pattern are almost sure to be bad. Consider that any creature, including man, is the product of millions of years of evolution. In every generation, those individuals with a gene pattern that fit them better for their environment won out over those with less effective patterns, won out in the race for food, for mates, and for safety. The more fit had more offspring and crowded out the less fit. By now, then, the set of genes with which we are normally equipped is the end product of long ages of such natural selection. A random change cannot be expected to improve it any more than random changes would improve any very complex, intricate, and delicate structure. Yet over the eons, creatures have indeed changed, largely through the effects of mutation. If mutations are almost always for the worse, how can one explain that evolution seems to progress toward the better, and that out of a primitive form as simple as an amoeba, for instance, there eventually emerged man? In the first place, environment is not fixed. Climate changes, conditions change, the food supply may change, the nature of living enemies may change. A gene pattern that is very useful under one set of conditions may be less useful under another. Suppose, for instance, that man had lived in tropical areas for thousands of years and had developed a heavily pigmented skin as a protection against sunburn. Any child who, through a mutation, found himself incapable of forming such pigment would be at a severe disadvantage in the outdoor activities engaged in by his tribe. He would not do well, and such a mutated gene would never establish itself for long. If a number of these men migrated to northern Europe, however, children with dark skin would absorb insufficient sunlight during the long winter when the sun was low in the sky and visible for brief periods only. Dark-skinned children would, under such conditions, tend to suffer from rickets. Mutant children with pale skin would absorb more of what weak sunlight there was and would suffer less. There would be little danger of sunburn, so there would be no penalty counteracting this new advantage of pale skins. It would be the dark-skinned people who would tend to die out. In the end, you would have dark skins in Africa and pale skins in Scandinavia, and both would be fit. In the same way, any child born into a primitive hunting society who found himself with a mutated gene that brought about nearsightedness would be at a distinct disadvantage. In a modern technological society, however, nearsighted individuals, doing more poorly at outdoor games, are often driven into quieter activities that involve reading, thinking, and studying. This may lead to a career as a scientist, scholar, or professional man, categories that are valuable in such a society and are encouraged. Nearsightedness would therefore spread more generally through civilized societies than through primitive ones. Then, too, a gene may be advantageous when it occurs in low numbers and disadvantageous when it occurs in high numbers. Suppose there was a gene among humans that so affected the personality as to make it difficult for a human being to endure crowded conditions. Such individuals would make good explorers, farmers, and herdsmen, but poor city dwellers. Even in our modern urbanized society, such a gene in moderate concentration would be good, since we still need our outdoorsmen. In high concentration it would be bad, for then the existence of areas of high population density, on which our society now seems to depend, might become impossible. In any species, then, each gene exists in a number of varieties, upon which an absolute good or bad cannot be unequivocally stamped. These varieties make up the gene pool, and it is this gene pool that makes evolution possible. A species with an invariable set of genes could not change to suit altered conditions. Even a slight shift in the nature of the environment might suffice to wipe it out. The possession of a gene pool lends flexibility, however. As conditions change, one combination of varieties might gain over another, and this in turn might produce changes in body characteristics that would then further alter the relative goodness or badness of certain gene patterns. Thus, over the past million years, for example, the human brain has, through mutations and appropriate shifts in emphasis within the gene pool, increased notably in size. End of section 6.